AP 120, Chapter 8, Topics, Signaling a Contraction and Contraction Cycle. So, we now know all the pieces to the story of how a nervous system signal leads to a muscular contraction. So now I'm just going to go through all the various steps and stages. This is a story, so try to remember it as such, with a beginning, a middle, and an end. And might not be as exciting as some stories out there, but it's the one we have. So to start with, we have the nervous system via the axon of a motor neuron will come down and form what's called a neuromuscular junction. So at the very end of the axon is a structure called a synaptic knob, and it lies next to a portion of the sarcolemma of the skeletal muscle fiber. And here's an awesome electron microscope view of the synaptic knob lying on the muscle fiber. Pretty amazing. And it's worth noting that our skeletal muscles are almost always voluntarily controlled, so they contract when we send that signal via the motor neuron. All right, so the signal arrives, and here is the synaptic knob. Here is the sarcolemma. Turns out the portion of the sarcolemma that is next to the synaptic knob is known as the motor end plate. So the synaptic knob of the axon of the motor neuron lies adjacent to the motor end plate of the skeletal muscle cell. So when we look really close, we see that within the synaptic knob are these vesicles. And these are called synaptic vesicles, and they are filled with a neurotransmitter called acetylcholine. Acetylcholine is a chemical that will help to signal for the muscle to contract. So within the synaptic vesicles of the synaptic knob, in the axon of the motor neuron, is found acetylcholine. Then it turns out there's a space between the neuron and the motor end plate, and that space is called the synaptic cleft. And then after the space, of course, is the motor end plate of the skeletal muscle cell. And what happens after the nerve impulse arrives is it triggers the release of acetylcholine. So the synaptic vesicles will migrate to the surface of the synaptic knob and will release acetylcholine into the synaptic cleft. So when the neural impulse arrives, acetylcholine is released into the synaptic cleft. Acetylcholine will then diffuse across the synaptic cleft and reach the motor end plate. And there are these transmembrane proteins found in the motor end plate that have binding sites or receptors for acetylcholine. So acetylcholine will bind to those receptors, and when they bind to the acetylcholine receptors, it causes those transmembrane proteins to open up and have open channels, allowing ions to flood into the skeletal muscle cell. So acetylcholine binds, ion channels open, and ions flood into the muscle cell. Now, to actually get a signal called a muscle action potential that leads to a muscle contraction, we need to reach threshold. To reach threshold, we need enough acetylcholine chemicals to be released and bind to enough of the receptors so that enough ions enter the muscle cell. So if threshold is reached, if that minimum stimulus is generated, then we generate a muscle action potential, shown by those dark arrows here. However, if not enough channels are open, then nothing happens. So either you get the response or you don't. It's an off-on switch. Now, the muscle action potential is a change in current caused by the opening of ion channels that moves down the length of the sarcolemma and into the transverse tubule. So here is the muscle action potential leaving the motor end plate, traveling down the sarcolemma, down into the transverse tubules. And as you remember, we have that triad. We have the sarcoplasmic reticulum adjacent to the transverse tubule. And when the muscle action potential that change in charge across the membrane caused by the movement of ions, when that reaches into the transverse tubule, that change in charge also affects the sarcoplasmic reticulum, and it causes the sarcoplasmic reticulum to release the calcium ions it is storing. And calcium ions are important for signaling the contraction. So again, the muscle action potential reaches the transverse tubule, and that signals the sarcoplasmic reticulum to release the calcium ions it is storing. So what happens next is, of course, happening on the molecular level, involving the proteins of the sarcomere. So the muscle contraction occurs at the molecular level via protein interactions between the thick and thin filaments. And this should be found on page 8.7. It's called the contraction cycle. So just a reminder, we have the thin filament, and the thin filament possesses the regulatory proteins tropomyosin. Tropomyosin covers a myosin binding site on actin, and also troponin. Troponin makes contact with tropomyosin and also possesses a calcium binding site. And then also found in the thin filament is actin. Actin possesses a myosin binding site. Actin is considered one of the two motor proteins. The other motor protein is myosin, which, as we know, makes up the thick filament. And myosin possesses an ATP-ADP binding site, so it needs energy, not a surprise for muscle contractions. And myosin is able to bind actin at that binding site. So here we have the thin and thick filaments. When there is no contraction, no signal, the muscle is relaxed, and we see that the thick and thin filaments do not touch. So then we have calcium released by the sarcoplasmic reticulum. This dramatic increase in calcium levels in the sarcoplasm leads to calcium ions binding troponin. Calcium ions bind troponin, causing troponin to change its shape. This then causes troponin to move tropomyosin. And by moving tropomyosin, this exposes the binding sites on actin. So again, calcium ions bind troponin. Troponin changes shape, moving tropomyosin and opening up the myosin binding sites on the actin proteins. So now we're ready for contractions because the myosin binding site on actin is exposed. So the next thing that happens is myosin binds to actin, and this forms a physical connection between the thick and thin filaments. This connection is called a cross bridge. So myosin binds actin, forming the cross bridge. Then myosin changes its shape. It moves, and as it moves, it causes the thin filament to get moved toward the end line. And this is the power stroke, sort of like trying to row, perhaps. So myosin strokes, myosin moves, and when it changes shape and moves, it moves the thin filament toward the end line. This then shortens the sarcomere. So this is when the sarcomere is shortened during the power stroke. Oh, wrong way. Then myosin releases ADP and phosphorus, and then it binds ATP. When ATP binds, that causes myosin to release actin. So we absolutely require ATP to bind to myosin in order to have myosin release actin to break the cross bridge. Then myosin uses the energy in ATP to return to its original conformation. So myosin uses the energy in ATP to reset, as it were. And then it either goes for another round of the contraction cycle. If there's plenty of calcium ions and plenty of ATP, myosin will just keep going along the contraction cycle, making the sarcomere shorter and shorter until it's as short as it can get. Or if the cell's running low on ATP, or if there's not enough calcium ions in the sarcoplasm, then the contraction cycle ends, and the muscle has the ability potentially to relax again. But as long as there's plenty of calcium ions, you will continue to get the contraction cycle occurring. Rigor mortis. Well, it turns out that rigor mortis is actually caused by this process, this contraction cycle. So initially, when someone dies, the body's warm, there's no stiffness in the muscles. Then a few hours later, the body starts to cool and starts to develop stiffness. And what's happening is that the sarcoplasmic reticulum is breaking down. And as it's breaking down, it's releasing calcium ions. And when enough calcium ions are released, this triggers the contraction cycle. So now you're getting some muscle contraction, some stiffness. Over time, though, this stiffness becomes even more powerful, more solid a muscle contraction. And this is because there is no more ATP being produced by the mitochondria. And as we know, myosin must bind ATP to release the thin filament. So if there's no more ATP, myosin is stuck attached to the thin filament, and this leads to what is basically a muscle cramp. And that is full-fledged rigor mortis. And then eventually over time, the stiffness will resolve because the myofibrils themselves start to break down. All right, oxygen supply and cellular energy. As we know, with oxygen present, we can get 36 ATPs produced from one molecule of glucose, which is pretty awesome. Muscles need a lot of oxygen because they need to make a lot of energy for muscle contractions. So initially they get their energy, the oxygen from hemoglobin that's carried to the muscle via the bloodstream. And then within the muscle's sarcoplasm, there is the protein myoglobin. Myoglobin very similar to hemoglobin, and it also binds oxygen. So oxygen is stored in the muscle fiber by binding to the protein myoglobin, and myoglobin just happens to be red, which is why muscles tend to be red. And now, after strenuous activity, it is possible to end up using up all of the oxygen within the muscle cell. And this then leads to fermentation, to anaerobic respiration, where you only get 2 ATP per glucose. And this is not ideal. It also leads to the production of lactic acid as the muscle cell ferments. The lactic acid ends up being returned to the bloodstream, where it goes to the liver. And then the liver will turn the lactic acid back into glucose. So oxygen deficiency is when the muscle fiber is out of oxygen, and so now it's using glucose inefficiently. And to try to get that glucose back, the lactic acid produced gets sent to the liver. Here is exercises. So low to moderate exercises, your muscle is going to be able to keep up, not a problem. High intensity exercises, eventually you'll run out of oxygen and have to go through fermentation. And eventually you'll just be exhausting the muscles. This is why people who lift heavy weights need someone to spot them so that when their muscles stop contracting, the weights don't fall on them. And that is muscle fatigue, losing the ability to contract during exercise. And this is caused by either decreased levels in ATP, as I just mentioned, or electrolyte imbalance, where the ions that are moving across the membrane to cause the muscle action potential are in the wrong ratios because the muscle has been signaled too many times in a row. And muscle cramps is when you have lack of ATP, and then you're unable to get the calcium ions back into the sarcoplasmic reticulum. That uses energy. Muscle fibers can't relax. And let's say muscle cramps, a lot like rigor mortis. So, boy, don't look forward to dying. Heat production, when muscles contract, they generate heat, and that is important for our body temperature. Also, cellular respiration, the making of ATP, also generates heat to keep our body temperature at 37 degrees Celsius. And that is it for this part of the lecture.